Hi folks, welcome to Bio 200 Genetics, and this is just an intro to genetics, chapter 1. We'll go through what you can expect this semester in this chapter 1. So first of all, this is called the central dogma of molecular biology, and that was coined by James Watson of Watson-Crick fame. James Watson and Francis Crick are the ones that discovered the double helical nature of DNA. And they coined the term, or James Watson coined the term, central dogma. What the central dogma says is that DNA only does two things. First of all, DNA is a template for its own synthesis, and that's called DNA replication. This happens during S phase of interphase in eukaryotic organisms. In prokaryotic organisms, they do not have a cell division cycle exactly the same, so we'll talk a little bit about that. The other uh, function of DNA is it's a template for protein synthesis. And protein synthesis is divided into transcription and translation, where transcription is copying the DNA template into an RNA molecule. It's also a nucleic acid, which we'll talk about, but it's slightly different. And then translation is decoding the nucleotide sequences into amino acid sequences of the protein. So the monomers of RNA are nucleotides, and the monomers of proteins are amino acids. And translation is converting from one code, the nucleotide code, to another code, the amino acid code. So this is the central dogma of molecular biology. And in one sense, molecular biology is very easy. DNA only does two things. But the devil's in the details, of course. And we'll talk a lot about the details of all of this. What genetics is the study of heredity, or how we transmit our inheritable information. Now, DNA contains our inheritable information, and genetics is the study of how we transmit that to offspring. So that's what we'll be studying. This is a DNA molecule. It's the molecule of life. We'll talk about how we came to discover it was the molecule of inheritance, starting from Fred Griffith in 1928 up through to Avery McLeod and McCarty in 1944. So we'll talk about this. When I was an undergraduate, we used to say we had 100,000 genes, and now we say we have about 25,000 genes. And we'll talk about why that is. So why in the 80s did we say we had 100,000 genes, and why today in 2018 do we say we have 25,000 genes? And we'll talk about that. So this is DNA. We'll talk about the details of this throughout this course. This is what we're showing you here is these are farm animals. Actually, they may not be. They may be cloned, but I'm thinking they're farm animals spelled that way. Farm, like pharmacology, pharmacy. And the reason why we'd call them farm animals is because the cells of the mammary glands right here will be uh, recombinant cells, meaning they probably have human genes in them, and then the milk from the goat will be producing a human protein. So we call them farm animals, the play on words, the pH of farm. This is copycat right here. Oh, actually, this one's carbon copy. We've cloned a couple cats. We have copycat, we have carbon copy, we have a couple other cats we've cloned. This is a cloned cat. Of course, we'll talk about Dolly. Dolly was the first cloned sheep. We'll talk about Dolly a little bit. And what you're seeing here in Time magazine are two very famous individuals. One is Craig Ventnor. This is Craig Ventnor right here. And this is Francis Collins right here. What happened was the U.S. government was doing the Human Genome Project, and the Human Genome Project was a project that was going to take about a decade, and it was going to determine the nucleotide sequence of the human genome. And there's about 3 billion base pairs in the human genome, haploid base pairs. That's really 2.9, but let me round it to 3. So there's about 3 billion base pairs. And if you don't know what a base pair is now, a base pair is when you have an A binding to T, that's one base pair, or a G binding to C in DNA. That's another base pair. And the human haploid genome has 3 billion base pairs. But we didn't know what the sequence was. We didn't know where the A's, T's, G's, and C's were. So now we do. And Francis Collins was spearheading this for the U.S. government, and Craig Ventnor was doing this in the private sector. And what happened was they decided to work together in doing this DNA sequencing. And because they decided to work together and share information, they finished the Human Genome Project 8 years before they were supposed to finish it, and it was a huge feat. We'll talk a lot about the Human Genome Project. Of course, it led to some other subdisciplines of biology. One big subdiscipline it led to is bioinformatics. You can get a degree in bioinformatics these days. Matter of fact, RIT, Rochester Institute of Technology, and many other colleges has a degree in bioinformatics. And what happened was with the Human Genome Project, the information, the raw data was tremendous, huge, and we needed computers to handle the raw data. And a new field called bioinformatics, which is really training people how to handle and parse this data. So you can have all the databases in the world, but if you don't have individuals trained to go in there and extract the data and do proper queries with proper whatever language you're using, then of course it's useless. So we have a new subdiscipline, several new subdisciplines, but bioinformatics was one of them. So we'll talk a lot about the discoveries in technology and what pushes the other, and we'll talk about that through this course. Genetic engineering, biotechnology, recombinant DNA technology. First of all, let me just be clear about something. Biotechnology is when you exploit an organism for human purpose. We have been doing this for thousands of years. Making beer is biotechnology. You exploit the yeast to make the alcohol. Making wine is biotechnology. Making bread is biotechnology. So biotechnology we've been doing for thousands of years. We exploit an organism for the human purpose. But it's only been recently that we have genetic engineering, where we go in there and we make the gene look the way we want it to look. And we'll talk about how we got there. And part of this genetic engineering and biotechnology is recombinant DNA. And what we can do now is we can take a human gene, say this is human cell right here. We can take a gene out of a human cell. We can ligate it or fuse it with some DNA from a bacteria. So this is a recombinant DNA molecule. Happens to be a plasmid of a bacteria. We'll talk about that. But it's part human and part bacteria. Put it back into the bacteria grows very quickly. It actually grows at 2 to the n, where if n takes 20 minutes, in 24 hours, n would equal 72. Well, 2 to the 72 is a tremendously large number. It's about 10 to the 21st. So we put this recombinant molecule in bacteria, let bacteria grow for about 24 hours. You have 10 to the 21 bacteria now. Therefore, you have 10 to the 21 recombinant DNA molecules. And then you just allow this gene to be expressed. And that could be human insulin, human growth hormone. It could be a gene for pesticide resistance. So now if you have corn that's pesticide resistant, you don't have to spray it with pesticides. It's already resistant by virtue of its own gene, or a recombinant gene in this case. You could put a gene in the bacteria that helps it digest oil better. I think the heart here is you could put a gene in bacteria that makes tissue plasminogen activator, TPA. Tissue plasminogen activator is a drug you can give to heart attack victims. That's a clot buster, basically. We'll go into some of the physiology of that later, but basically bust clots. If the gene was human growth hormone, you could cure pituitary dwarfism. Now, why is this important? Well, back in the day, for example, diabetics, they were taking pig insulin, porcine insulin. Well, the insulin is the insulin, and it helps us, the diabetic, to lower blood sugar, but they could react to the pig insulin. It's a foreign protein to them. They could have an allergic reaction to it. But if it was human insulin, made by recombinant DNA technology, then they're going to accept that and not react to it. It's human insulin. So you could have human insulin here being made to treat diabetes. And the list goes on and on, but that's where we're going with talking about genetic technology. We can actually take some genes out of some organisms like jellyfish, and that particular protein is called green fluorescent protein. It comes from a jellyfish. There's other bioluminescent genes, by the way. When I was a graduate student, I worked with Vibrio fischeri, and that has a gene in it called, well, an operon in it, a set of genes called the Lux operon. And the Lux operon is a set of genes that lead to chemiluminescence, or it's bioluminescence also, because it is a living thing. So this is a bioluminescent gene. Hopefully I spelled that right, bioluminescence. And I worked with this. I was determining how close the firefly luciferase gene was to the Vibrio fischeri Lux operon. And it turns out that one of the genes of the Lux operon is very similar to the luciferase gene of fireflies. So this green fluorescent protein is a fluorescent molecule coded for by a gene. And we can take this gene and we can put it into, say, a mouse. And now that mouse will glow green. We can take the green fluorescent protein and stick it in the gonads of, what is this? It's the larva of a mosquito. So this is really a reporter construct. We use this green fluorescent protein to tell us where things are, or are they there? Did we successfully get it there? Well, how do we know we successfully got it there? It glows green. That's pretty elegant that we used green fluorescent protein as a reporter construct to tell us if the gene got there, because you can see it. So we have that we'll talk about. This is showing you from the central dogma up to applications. So remember what the central dogma is. It's DNA does two things. DNA is a template for its own synthesis, and it's a template for protein synthesis. From the central dogma, you see the DNA coding for proteins here. Well, you can study the DNA, and that's called genomics. Studying all the genes of an organism is the genome is all of the genes the organism has. That's called genomics. We can study it from the protein chemistry viewpoint, and that's called proteomics. That's all the proteins in the cell. That would be the proteome. The genome is all the DNA in the cell. The proteome is all the proteins in the cell. We can use the information from this to do different things like make clones. We can make reagents like PCR primers. You might not know what PCR is. I'm just giving you some examples to sink your teeth into it. And then we can apply this. Apply this to what? We can apply it to agriculture. We can make plants pesticide resistant without spraying them with pesticides. We can make human growth hormone and cure pituitary dwarfism. We can make human insulin and treat diabetes, et cetera, et cetera. So you can see that this just has huge impacts on science and humans in general. And then down here is a timeline. We'll talk about Gregor Mendel. He is an Austrian monk that worked with pea plants and showed really the father of genetics, and showed how an inheritable unit of inheritance exists. Now we didn't know that the unit of inheritance was a gene, which is made of DNA. We had no idea back then, but we'll talk about how he discovered that there was a discrete unit of inheritance back in the 1860s. We'll talk about Fred Griffith in 1928, right around in here. His transforming principle was DNA. He didn't know it at the time, but it was. We'll talk about Avery, McLeod, and McCarty right in here. They demonstrated that the transforming principle was DNA. We'll talk about Watson and Crick, double-stranded nature of DNA. We'll talk about elucidating the genetic code in the 60s. We'll talk about Hamilton Smith and restriction enzymes starting in the 70s, and the whole revolution of molecular biology up to Kary Mullis in the 80s and PCR. And we'll just talk about all these throughout the whole semester. So this is not a slide where you have to sit here and start memorizing these things. I'm just telling you where we're going with all of this, and that's what we'll be talking about. Let's take a look at molecular expression. And again, I'm really jumping ahead to a future chapter, but it's all based on this. It's based on the readout of DNA. So let's just take a look.
The sequence of nucleotide bases in DNA carries genetic information in units that are called genes. Structural genes encode the information for specific proteins. To create a protein, a gene must first be transcribed into a sequence of nucleotide bases in a messenger RNA molecule. The messenger RNA sequence is then translated into an amino acid sequence of a protein. This sequence of amino acids in a protein molecule determines the shape and chemical characteristics of the protein. Thus, each structural gene specifies a specific protein in the cell that carries out a specific function based on its chemical characteristics and molecular shape. This function of the specific protein gives the cell and the organism the specific trait coded by the gene.
Okay, so that's the central dogma, a part of the central dogma. Remember, part of it is DNA is a template for its own synthesis. But this is the other part of it where it says that DNA is a template for protein synthesis. All right, let's talk about some nomenclature. So what is a characteristic? Well, a characteristic and a trait, they sound similar, and they are similar, but it's more than a subtle difference. A characteristic is a variable like height. So you could say a characteristic is height. The trait is a variation on that variable, like short or tall. So you have a gene, if this is a chromosome, this is a loci or an area on the chromosome containing a gene. Here's your homologous chromosome. Here's the loci that goes with it. These homologous loci right here, one came from your mom and one came from your dad, they don't have to code for the same trait. One could say tall and one could say short, and I use little t for short. But by the way, height doesn't follow simple Mendelian genetics where one gene is completely dominant over another. I'm keeping things simple for now, so I don't want you to get the idea. But the characteristic is the variable, height or pigmentation, etc. And then the trait is what version of that variable do you have? Are you tall? Are you short? So that's the difference between a characteristic and a trait. We have different types of traits. We have morphological traits. These are the kind of traits that you think about. Is it a red flower or a white flower? These are morphological traits. We have physiological traits. This is basically, do you have the enzyme or not have the enzyme? And it doesn't have to be always have to be an enzyme either. It could be a particular cell with a particular cell surface protein. So physiological traits are things like, do you have the enzyme? Enzyme for what? Well, enzyme to convert alcohol to acetaldehyde. Alcohol to acetaldehyde. Some people don't have this enzyme. That means they don't metabolize alcohol very quickly. The name of the enzyme is alcohol dehydrogenase. So some people have alcohol dehydrogenase and some people do not have alcohol dehydrogenase. So this is a physiological trait. And then we have behavioral traits. Behavioral traits are more complex because some behaviors they're all based on, say, a simple reflex arc, but there are some complex reflex arcs. So behavioral traits start with, say, some reflex arcs, and they go up into some very complex events involving your limbic system of your brain and things like that. The drive and motivation for why you do what you do. So we can talk about traits based on morphological traits, physiological traits, or behavioral traits. I'll tell you one very cool thing in biology is there's some behavioral traits in very young organisms, babies, and just juvenile organisms. And these traits are supposed to go away as the organism matures into an adult. And there's a phenomenon called pedomorphosis. Pedomorphosis. This is retaining your juvenile behavioral trait into adulthood. It actually could be retaining a juvenile morphological trait into adulthood as well, by the way. It doesn't have to be a behavioral trait. But the reason why I'm using this in the behavioral trait is because there is I want to say it's a lizard there's a lizard that when it's born, it has a behavioral trait where it holds perfectly still when there's an environmental threat. But that behavioral trait goes away as the lizard matures into adulthood. Now, I think the biological idea behind that is that the mother will draw the threat away from the baby and the baby holds perfectly still. You see this in white-tailed deer as well. A fawn will lay right down and hold perfectly still, and the mother will take off and draw the coyotes towards her, and the baby will be safe, hopefully. With this lizard, the same thing happens. The baby lizard will hold perfectly still, and the mom lizard will take off and draw the threat towards her. But there has been a phenomenon in this lizard species where a pedomorphotic phenomenon where the adults start to lay down perfectly still in in the face of an environmental threat. Well, as it turns out that doesn't work very well because these very aggressive carnivorous ants will be attacking the lizard, and instead of the lizard running away from them like it can, it lays down and the ants have a smorgasbord. So obviously that wouldn't last very long. It wouldn't be selected for, but we have seen that in biology. So we have these different types of traits. Now, when you start thinking about genetics and the transmission of inheritable units of heredity like genes, and we start talking about dominance and dominant and recessive and codominant and incomplete dominance and epistasis and things like this, you have to remember it always depends on at what level you interrogate the system. For example, you could have big B is dominant over little b, which is recessive. All right, so big B for brown, little b for white. Now, if you look at the molecular level, if you have little b, do you make the protein? This is little b, this is big B. Well, at this level, the answer is yeah, you make the protein. Now, it happens to be poorly functional, but you make it. So at the molecular level, these are codominant. If you have the big B gene, you make the pigmentation enzyme, and if you have the little b gene, you make the pigmentation enzyme. As it turns out, the little b gene codes for a poorly functioning enzyme, but you still make the protein. So at the molecular level, these are codominant. Then you look at the cellular level, and at the cellular level, they look to be maybe a little bit incompletely dominant. Because when you have the big B gene, you make a lot of melanin, or whatever the pigment molecule is, trichocydrin, whatever the pigment molecule is. But when you have the little b gene, you make very little pigment. So this could even be argued, depending on how little is little, this could be argued to be a dominant recessive relationship, maybe an incomplete dominant recessive relationship. Depends on if the heterozygote actually has a different phenotype than the homozygote. That's what it really depends on. And then at the organism level, if you have the big B gene, you are a dark butterfly. And if you have the little b gene, you are a white butterfly. What if you have the big B little b gene? Well, if you have the big B little b gene and you are just like the big B, then that is a completely dominant recessive relationship. But if the heterozygote is someplace in the middle between homozygote dominant and homozygous recessive, then you might have some incomplete dominance there. And then at the population level, this is where environmental stresses select. Selective pressures select at the population level. And what you can see here is the dark butterflies are more fit in forested regions because they'll land on a dark area of the tree and be hidden from the bird that wants to eat it. Whereas if the white butterflies were to land on a dark tree, it would show up very clear for the bird, and they wouldn't be as successful in the forest. But the light butterflies, they're very successful in the meadows and fields, unforested regions, because they have light areas to land on and the birds can't see them. So at the population level, some genes will have advantages to other genes, and we'll look at these selective pressures. And of course, that has everything to do with natural selection and evolution, and we'll talk about that. So remember, how we define a gene, whether it be dominant, recessive, incomplete dominant, codominant, epistatic. How we define a gene depends really on what level of the system you're interrogating the system. Are you interrogating at the molecular level, the cellular level, the organismal level, or at the population level? What are you really looking at? And I'll keep reminding you of this as the semester unwinds. We have morphs in populations, and what a morph is within a given species. These frogs can mate, can interbreed. They can mate and have fertile offspring. So they're the same species. We have very contrasting forms within a species, and these are called morphs, morphological differences, or morphs. A lot of times, scientists will incorrectly misclassify these guys because they look so different. But you do the DNA analysis now, and it tells you a lot about them. So we do have some genetic variation with species that lead to morphs or drastic differences. Some other sources of genetic variation. Well, first of all, we have gene mutations. We'll talk a lot about that. That's a nucleotide change in a gene. We have changes in chromosome structure. You can have translocations and deletions and additions in chromosome structure. You have changes in chromosome number. That's what this picture is showing. This girl right here, she has trisomy 21. She has 3 number 21 chromosomes. That's called Down syndrome or trisomy 21. That's a genetic variation. The average human being has 2 number 21 chromosomes. This Downs athlete here, she has 321 chromosomes. I don't know what the wheat is showing you. It's probably polyploidy. I'd have to look at the book and see what it's showing you, polyploidy. But plants do this very unique thing that they can become polyploidy. And polyploidy, it can be an advantage for some fruiting plants, some angiosperms, and we'll talk about some of that. So there's lots of different things that add to our genetic variation, and we'll talk about all of that. There's environmental influence on your genes. Hydrangeas, and some, not just hydrangeas, but other flowers as well. They can be genetically identical, but based on the pH of where they grow, their flower can be different colors. So this plant isn't genetically identical to this plant, but look at their flower colors. Well, how could that be? Well, their environment plays a role, and the pH of the soil actually plays a role. You can see if the pH of the soil is very low at 4.5, you get a deep blue. But if the pH is neutral, you get pink. So with a given genetically identical flower, the environment plays a role on the readout of the genes. So we'll talk about nature versus nurture and the environmental role in gene readout. This is another example of environmental role. We have a disease, humans have a disease called phenylketonuria. PKU, if you look at any diet soda, Diet Coke, Diet Pepsi, it'll have a warning on it. It'll say, caution, phenylketonurics contains phenylalanine. Well, yeah, you're lacking this enzyme right here. Phenylalanine hydrolase, you're lacking it. Well, when you lack the enzyme, phenylalanine is converted into these toxic products that together can lead to mental retardation and even death and developmental abnormalities. And so how do you prevent that? Don't eat phenylalanine. Simple. Don't eat phenylalanine. You grow up to have a nice long life. Well, to not eat phenylalanine, you have to know which foods have phenylalanine in it, and you have to not eat it. So there you go. You limit your intake of phenylalanine, and you can have a normal life. And that's where your environment, how much phenylalanine you take into your body, affects the readout of your genome. Now, what genetics is all about, of course, is transmitting the unit of inheritance, the gene. And Gregor Mendel is the father of genetics, and he studied this in pea plants. We'll talk a lot about that. He did this in the 1850s. It's saying mid-19th century. That's the 1850s. We'll talk about him quite a bit. This is showing you actually an artistic drawing of, say, a karyotype. What the human genome, what we do with it is we take the longest chromosome, and by convention we call it number 1. We take the shortest chromosome, and we call it number 2. And we just line them up from tall to short, from 1 to 22. And then we have and those are autosomes. 1 to 22 are autosomes. They are chromosomes not involved in sex determination. And then you have your sex chromosomes. If you're female, you're XX. If you're male, you're XY. This is showing you a haploid cell in a human. doesn't have homologous chromosomes. Homologs are uh, chromosomes where one came from your mom and one came from your dad. So when you're haploid, you don't have homologs, no homologs. Your eggs and sperm are haploid, no homologs. Whereas if you have a diploid cell, you have homologous chromosomes. So for this chromosome number one came from your dad and one came from your mom. From this chromosome number two, one came from your dad and one came from your mom, and so on and so forth. So this is a diploid cell who has homologous chromosomes, and this is a haploid cell who does not have homologous chromosomes. And we'll talk about reproduction and making haploid gametes, that means eggs and sperm, and fertilization and passing your DNA on to your offspring. We'll talk about evolution quite a bit. Evolution is nothing more than DNA changing over time. I'll probably have no issues talking about microevolution to the class. Oh, can't spell. Usually microevolution makes perfect sense to everybody, but it'll be macroevolution where there might be some pushback. I'm not here to propagandize anybody. If you have a personal reason why you are against the theory of evolution, I respect that. But this is a science class, and right now evolution is the best story going to explain the unity and diversity of life. So, and there's a lot of data to support evolution. We'll talk about the data, and then you can make your own decision. We'll talk about is it a perfect story? Nope, there's some missing pieces, but it's the best story going to explain unity and diversity of life. So we'll talk about evolution, and we'll talk about both microevolution and macroevolution. What you see here is macroevolution from the common ancestor of all horses. So how did this little animal right here become this big horse right here? Well, that's macroevolution. But within that, these little small changes, in a couple hundred years or so, that's all microevolution and gene frequency changes generation to generation. So we'll talk about both in this course. We'll talk about natural selection, which is environmental pressures on organisms that drive gene frequency changes, and basically it's an environmental pressure that selects for a certain gene and selects against another gene. And what I'll do is I'll say, well, humans have applied artificial selective pressures on different organisms. This is wild mustard, Arabidopsis, or yeah, Arabidopsis. Well, I actually don't know if wild mustard is technically Arabidopsis, but it's in the same. Yeah, it's in the same. It's in the same genus. Oh, I forgot how to spell Arabidopsis, so forget that. But this is wild mustard. Brassica. No, it's not Arabidopsis. It's Arabidopsis. It's Brassica. Boy, I wish it told me. Anyway, whatever the genus of wild mustard is, humans have applied artificial pressures on wild mustard to make these foods we eat. It's just what these foods are different parts of the plant that have been exaggerated. Like kohlrabi is an exaggerated lateral meristem. Kale is just an exaggerated leaf of brassica. Cabbage is a really short internodal length. That's what cabbage is. It's a very short internodal length. Broccoli, what that is no flowers. Or well, I shouldn't say no flowers, because of course you have to have flowers to reproduce, but suppression of the floral development. And cauliflower, you have the flowers, but they're sterile. So this is selective pressures, artificially man-made pressures on wild mustard to give us a lot of the vegetables we eat. We've done the same thing with our dogs. All of our tame dogs come from a wolf, and it's been artificial selective pressures that give us our different dogs. And I don't know what all these dogs are right here. You guys probably know better than I do, but here are different types of dogs. Like I'll take a chance that someone's going to yell at me. Like this ugly little thing right here. That came from a wolf, just like this dog came from a wolf. All right, so artificial selection, natural selection. Then we'll talk about different fields of genetics. We'll talk about model organisms. We'll actually talk about that very shortly. Like Cyanorhabditis elegans, it's a little nematode, a little roundworm, like Drosophila, fruit fly. We'll talk about some other one. E. coli, that's probably number one. A lot of what we know comes from the bacteria Escherichia coli. There's some other ones. We'll talk about transmission genetics, and the transmission genetics are these chapters. We'll talk about molecular genetics. That's my favorite area. Molecular genetics is those chapters. And then lastly, this is also, it's not my favorite, but I like it a lot. Population genetics are the chapters 25 and 26. We'll talk about Hardy-Weinberg and some mathematical models of looking at gene frequencies. We'll do some binomial expansions, and I'll remind you what they are in case it's been a while since you had statistics. So that's where we're going with this. Here are some model organisms. So here's C. elegans right here. I worked with C. elegans as a graduate student. Here's E. coli. I worked with E. coli as a graduate student. We still work with E. coli today in microbiology. This is Saccharomyces cerevisiae. This is just baker's yeast. You can go to Walmart and buy this Saccharomyces cerevisiae. It's just yeast. This is Drosophila, a fruit fly. Back when I was an undergraduate, we had fruit fly labs in our genetics class, and of course you put them to sleep with this stuff called fly nap. It's really a tetrasubstituted ammonium. What tetrasubstituted means is there's different chains coming off the nitrogen compound, so it's tetra four substituted units, and FlyNap puts the Drosophila asleep, but they would wake up before you were able to completely look at them with a magnifying glass or dissecting microscope, and they'd get away, and then pretty soon you had, because they reproduced, you would have fruit flies all over the lab. Mouse. One of my graduate advisors did a lot of endocrinology on mice. I didn't really work with them much other than to feed them sometimes for him. This is a Arabidopsis thaliana. It's related to Brassica. In other words, it's related to wild mustard, but obviously since it's not named the same, it's slightly different. I gotta admit to you that I'm not a botanist, so I'm gonna have to go look that up later and see how true what I'm telling you is. But there's a Arabidopsis, very fast growing plant. So those are some model organisms that we use in genetics. Okay, the science of genetics. Let's talk about, there's two different types of testing. We have hypothesis testing, which is investigative testing. You're actually investigative testing. You're actually manipulating something to see how it turns out. Or you have discovery-based science, and it's just observational. I mean, might have to manipulate it to measure something. You can't see how many chromosomes are in a cell. You have to manipulate it to stain them to see it. But it's really just observational. You're just seeing. You look at different cancers and say, is there anything the same in all these cancers? So it's just looking at them. You're not making a hypothesis. You're not doing a controlled experiment. You're just saying, hey, I wonder if there's anything the same in all these cancer cells. Something like that is going on. So we have hypothesis or investigative testing, and we have discovery-based science. Both are very important. This is the scientific method. It's not something you can't deviate from. I don't want you to think it's written in stone and you can't deviate. You of course can. But first you make an observation, like this lady made an observation. Her flashlight didn't work. And she asked herself a question. Well, I wonder why the flashlight doesn't work. Could it be the batteries? Could it be the bulb? Well, those are hypotheses. Your best guess, an educated guess, if you will. And then what you do is, if you say to yourself, well, I think hypothesis 1 is true, then you're going to replace the batteries. That's your prediction that it's the batteries. And then you put the batteries in and you test the flashlight. Turns out you have brand new batteries in there, assuming you put them in there oriented correctly, and the flashlight still doesn't work. Well, that hypothesis was demonstrated to be false. Your data did not support the hypothesis. So you go to the next hypothesis. You say, well, my light bulb must be bad. You get your new light bulb, you unscrew it, you put it in there, then you test your hypothesis, and your flashlight works. So now you just threw out some good batteries. Now you're going to say, you might say to yourself, well, could it be both? Yeah, it could be both. But typically speaking, we do one hypothesis at a time. We do it in a very stepwise fashion. You don't sit there and change 4 variables, because if you do, then you never knew which one was wrong. If I changed the bulb and the batteries at the same time, I didn't know which one was broken. But if I do them one at a time, then I figure out what was wrong with my flashlight or my system. So this is the scientific method or the scientific process. Interpreting data, we're going to look at a lot of graphs. I'm not going to explain this to you in great detail right now, but like this is a graphical view of what's going on here. And what's going on is we have a conjugation, a sex pilus or a conjugation pilus right there, and some DNA from this bacteria is going to go into that bacteria. That's what's going to happen. And what we can do is we can see, well, what order does the DNA come through? And we can blend this up at 5 minutes, 10 minutes, 15 minutes, 20 minutes, and that blending breaks this conjugation bridge. But if I break it at 5 minutes and I find out that I've only transferred the pro-AB gene, then I know that the pro-AB gene is at the beginning of the origin of replication. And that's one of the first genes to go in. Whereas if I look what was transferred after 10 minutes and I find that the lock operon was transferred, pro-AV and lock operon was transferred after 10 minutes, then I know that the lock operon is the second set to go through. And I can actually map my DNA by doing this kind of experiment. And you can graph and see what happens at different times of blending. So if you blend at 16 minutes, the only thing that went in was LAC-Z, Gal-E didn't go in. But if you blended at 25 minutes, then LAC-Z and Gal-E went into the recipient cell. So you can map your DNA, and we can look at graphs to see what's going on. So we'll do a lot of interpreting of data. We'll come back to this picture. It's a future chapter, and we'll talk a lot about it. That's it, folks. Welcome to genetics. That's chapter 1. Just a quick overview, or maybe not that quick. And see you in chapter 2.